Yes, good evening, dear students. Welcome to Sankalp learning classes. Already we have completed the chapter that is principles of inheritance and the variation. So inheritance, what is the meaning of inheritance? So that already we have studied in the last class. So in this chapter, we are going to study about the molecular basis of the inheritance. So mainly about the DNA and the RNA, DNA and RNA, very importantly, and DNA replication and synthesis of the protein and discovery of the DNA and characteristics of the DNA. So these all things we are going to study in this chapter and about the RNA. Okay. So DNA. So Mendel's used the word factors for these genes, right? The factors which are transferred from parents to the next generation. So he used the word factors. Now we are using the word gene. Okay, so gene, that is nothing but the segment of the DNA. Gene is nothing but the segment of the DNA. So this DNA is responsible for the deoxyribonucleic acid. So this DNA is responsible for the transmission of characters from parents to the next generation. So ಮೆಂಡಲ್ಸ್ ಏನ್ ಸ್ಟಡಿ ಮಾಡಿದ್ರು, ಸೊ ಅವರು ಫ್ಯಾಕ್ಟರ್ಸ್ ಅಂತ ಒಂದು ವರ್ಡ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿದ್ರು ಅಲ್ವಾ. ಸೊ ಈ ಫ್ಯಾಕ್ಟರ್ಸ್ ನೌ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೀನ್, ಅಂಡ್ ಜೀನ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಸೆಗ್ಮೆಂಟ್ ಆಫ್ ದ DNA. ಇಲ್ಲಿ DNA, so that is the genetic material in most of the organisms, and RNA, that is also genetic material, and it is also act as the catalyst and the adapter molecule. So RNA is also a genetic molecule in lower organisms like viruses. Okay, so let us study about this DNA and RNA in this chapter. So the DNA, that is the polymer of deoxyribonucleotides. So what is this deoxyribonucleotides? So that we learn in the next slide. Okay, the length of the DNA depends on number of nucleotide pair present in it. The length of the DNA. So what is the length of the DNA? That is present, that is depends on the number of nucleotide on the present in the DNA, and characteristics of this organism depend on the length of the DNA. So the DNA is responsible for the characteristic feature of the organisms. And here bacteriophage, so that is having the 5386 nucleotides, and bacteriophage lambda has the 48,502 base pairs. So that is the length of the DNA in this bacteriophage, that is the virus. And E. coli bacteria have the 4.6 into 10 raised to 6 base pairs, and human genome, that is haploid. So haploid genome has the 3.3 into 10 raised to 9 base pairs. Just this is the some basic information about the DNA. Okay, then let us see about structure of polynucleotide chain. Here we use the word polymer of deoxynucleotides, right? So what is the meaning of nucleotide and polynucleotide? Let us study about that. A nucleotide has 3 components. Okay, so nucleotide which is present in the DNA. ಈ DNA ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, DNA that is made up of the nucleotide. Okay, DNA ಅಂದ್ರೆ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, ಜಸ್ಟ್ ಯಾವ ತರ ನೀವು ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ತೀರಾ ಈ ರೀತಿಯಾಗಿ ಅಲ್ವಾ. ಸೊ ಈ DNA that is made up of the nucleotide, and nucleotide, so what is nucleotide? This nucleotide, that is a nitrogenous base, and the pentose sugar, ribose in case of the ribose sugar in case of RNA, and deoxyribose sugar in DNA. Okay, and phosphate group. So the nucleotide that is having the nitrogen base and pentose sugar and the phosphate group. So these all together that constitute the nucleotide. Then what is this nitrogen base? There are two types of the nitrogen base, purines and the pyrimidines. Purines are adenine and guanine. Okay. There are mainly two types of the nitrogen base. Nitrogen base ಅಲ್ಲಿ two types ಇದೆ, purines and the pyrimidines. Purines that is adenine and guanine, and pyrimidines cytosine and the thymine. Uracil that is present in RNA. So instead of thymine, okay, cytosine ಮತ್ತೆ thymine so ಎಲ್ಲಿರುತ್ತೆ DNA ಎಲ್ಲಿರುತ್ತೆ, and uracil that is present in the cytosine and uracil that is present in the RNA. Okay, so that is the purines and the pyrimidines. They are the nitrogen base. Nitrogen base, purines and pyrimidines. Then this nitrogen base is linked to the OH group of the first carbon of the pentose sugar through the N-glycosidic linkage. Okay, I will show this using the diagram. Yes, this is the, okay, so this is the nitrogen base, adenine. Just imagine this is the adenine or any nitrogen base. So that is attached to the pentose sugar. So this is the pentose sugar at the first carbon of the pentose sugar. Okay. By the linkage. So this is the bond between this pentose sugar and the nitrogen base. Nitrogen base ಇಲ್ಲಿ ಅಡಿನೈನ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, ಯಾವುದೇ ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಇರಬಹುದು. ಅದೇನಾಗುತ್ತೆ, ಸೊ ದಿಸ್ ಇಸ್ ದ ಶುಗರ್, ಪೆಂಟೋಸ್ ಶುಗರ್ ಗ್ರೂಪ್ ಗೆ ಏನಾಗಿದೆ, ಫಸ್ಟ್ ಕಾರ್ಬನ್ ನಲ್ಲಿ ಅಟ್ಯಾಚ್ ಆಗಿದೆ ಅಲ್ವಾ. ಸೊ ಥ್ರೂ ಎ ಬಾಂಡ್ ಎನ್ ಗ್ಲೈಕೋಸೈಡಿಕ್ ಬಾಂಡ್. ಇಲ್ಲಿ ನೋಡಿ N-glycosidic linkage to form the nucleoside. So here nitrogen base plus pentose sugar, it is called as nucleoside. Nitrogen base ಏನಾಗಿದೆ, pentose sugar ಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದೆ, ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ಇನ್ನೂ ಅಟ್ಯಾಚ್ ಆಗಿಲ್ಲ ಅಲ್ವಾ. ಸೊ ಅದಕ್ಕೆ ಇದನ್ನ ಏನಂತ ಕರೀತಾರೆ, ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಅಂತ ಕರೀತಾರೆ. such as if adenine is attached to the pentose sugar, then it is called as adenosine or deoxyadenosine. If the guanine is attached to the pentose sugar, then guanosine or deoxyguanosine. If the cytidine is attached to the pentose sugar, so deoxycytidine or cytidine. If uracil is attached to the pentose sugar, it is called as deoxythymidine. Uridine or deoxythymidine. Okay, so this is the nucleoside. So how it becomes nucleotide? So nucleotide means it contains the nitrogen base, pentose sugar as well as phosphate group. So the phosphate group is linked to the OH group of the 5' carbon atom, 5th carbon atom of the nucleoside through the phosphodiester linkage. Corresponding nucleotide, so that is depending on the type of sugar present in it, and the nucleotides are, yes, let us see the diagram. Yes, so this adenine is attached to the pentose sugar. Okay, only adenine, no phosphate group. Then it is called as nucleoside. Here, this nucleoside is called as adenosine or deoxyadenosine. Guanine or deoxyguanosine. So like this it is called. If the phosphate group is attached to the 5th carbon atom of this pentose sugar through a phosphodiester bond, then it will become the ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ಹಾಗಾದ್ರೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲಿ ಏನೇನಿದೆ ಅಂತ ಆಯ್ತು? ಸೊ the nucleotide it contains, please comment in the comment section. Yes, it contains the nitrogen base, adenine, thymine, guanine, or cytosine or uracil in case of the RNA, and pentose sugar, deoxyribose sugar, okay, and phosphate group. Together it forms the nucleotide. So this is the one nucleotide. Here the nucleotides, two nucleotides are linked through the 3' to 5' phosphodiester linkage to form dinucleotide. More than more nucleotides can be joined in such manner to form the polynucleotide chain. So this is the meaning of the polynucleotide chain. Here this is one nucleotide, right? And this nucleotide that is attached to the another nucleotide through a 5' to 5th and 3rd phosphodiester linkage. So ನೋಡಿ ಇಲ್ಲೆಲ್ಲ ಏನಾಗಿದೆ, ಥ್ರೂ ಫಾಸ್ಫೋಡೈಎಸ್ಟರ್ ಲಿಂಕೇಜ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಜಾಯಿನ್ ಆಗಿದೆ ಅಲ್ವಾ. ಈ ತರ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಜಾಯಿನ್ ಆಗಿ ಏನಾಗುತ್ತೆ, ಸೊ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಚೈನ್. So that is the meaning of polynucleotide chain. Okay. Then polynucleotide chain has free phosphate moiety at the 5' end of the sugar. So is referred as 5' end. In the other end, the polymer with 3' OH group is called as the 3' end. Okay. Here it has the phosphate group at one end, 5' phosphate group at the one end, and here OH group at the another end. So it has the polarity. So this is called as 5' to 3' polarity. Okay. So that is called, the nucleotide chain has the 5' to 3' polarity. The backbone of polynucleotide chain is sugar and the phosphate. Nitrogen bases linked to the sugar moiety project from the backbone. Main backbone of this polynucleotide chain is sugar and the phosphate. In RNA, every nucleotide has additional OH group at the 2' of the second carbon of the ribose. So in uracil, in RNA, uracil is found in the place of thymine. So that we know. In case of RNA, uracil is found in case of the thymine. Okay, so that is the pyrimidine. So that is about the polynucleotide chain of the DNA. I hope this concept is clear. Yes, this is the phosphate group and this is the sugar, deoxyribose sugar in case of DNA, and this is the nucleotide, nitrogen base. Together it constitutes the nucleotide. Okay, so mainly the nucleotide has the 3 components: a nitrogen base, a pentose sugar, and the phosphate group. Nitrogen base, they are purines and pyrimidines. Purines ಯಾವ್ಯಾವುದು, pyrimidines ಯಾವ್ಯಾವುದು ಅಂತ ನಿಮಗೆ ಗೊತ್ತಿದೆ ಅಲ್ವಾ. So that forms the nucleotide. A nucleotide join continuously, more nucleotides join together and form the polynucleotide chain. Let us study about the history of the DNA, how it is discovered and how it is first observed by the scientists. So that we are going to study in this part. DNA is an acidic substance in the nucleus, was first identified by the Friedrich Mischer in 1869. He named it as a nuclein. Here the DNA is acidic in nature, acidic substance, okay, mainly found in the nucleus, and it is first observed by the scientist Friedrich Mischer. Okay, but he named it as not a DNA, he named it as nuclein. Okay, but in the 1953, so he observed that in 1869, so in 1953, double helix structure of DNA was given by James Watson and Francis Crick based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. Here, this double helix model of DNA. So the structure of DNA, how it is. Okay, the double helix model of very famous theory of the the famous theory that explains the structure of DNA, that is the double helix model of DNA, that is given by the scientist Watson and Crick. Okay, here James Watson and Francis Crick, and it is also called as Watson-Crick model of DNA. Then how it is discovered? Mainly the X-ray diffraction data that is produced by the Maurice Wilkins and Rosalind Franklin. They collected the data from their experiments and proposed the theory that is called as the double helix structure of DNA, and also called as Watson and Crick model of DNA. Hallmark of that proposition was based on pairing between the two strands of polynucleotide chain, and this was based on the observation of Erwin Chargaff. So the scientist, the Chargaff observation, what is that? Chargaff observed that the double-strand DNA, the ratio between the adenine and thymine and guanine and cytosine are constant and equal 1. So very important. This is the Chargaff rule. Yes, let us see the silent features of double helix structure of DNA. Here, the DNA is made up of two polynucleotide chain. Means this is just, I will draw like this. This is one polynucleotide chain. Nucleotides are joined. And here, another polynucleotide chain. Okay. DNA is made up of two polynucleotide chain. And sugar and phosphate forms the backbone and bases projected to inside. Two chains have anti-parallel polarity. You know that these polynucleotide chains, they have the polarity. So here 5 prime to 3 prime. If one polynucleotide chain is having the 5 prime to 3 prime polarity, and another chain that is having the anti-parallel polarity, means it is having the 3 prime to 5 prime polarity. This chain is having the 3 prime to 5 prime, that is anti-parallel polarity. Two strands are held together by hydrogen bond present between the bases. Here, these two strands are held together by the hydrogen bond. So, hydrogen bond is present in between the two strands of the polynucleotide chain. Here, always in the DNA, adenine, the nucleotide which contain the adenine, pairs with the thymine, always AT and C, cytosine and the guanine, ATCG. Adenine always pairs with the thymine with a 2 hydrogen bond, and cytosine, guanine of one strand pairs with the cytosine of another strand by 3 hydrogen bonds. Here, adenine and thymine, so they are bound by 2 hydrogen bonds. So these are the nitrogen base present in the polynucleotide chain, and cytosine and guanine, they are paired with they are joined by 3 hydrogen bonds. So this is very important. So how many hydrogen bonds are present in the DNA in between the nitrogen bases? So this is the silent features of the double helix model of DNA made up of two strands. Yes, why two strands are arranged in anti-parallel. So so that is the basic structure of DNA. So DNA ಅರೇಂಜ್ ಆಗಿರೋದೇ ಹಾಗೆ. ಹೇಗೆ ಅರೇಂಜ್ ಆಗಿರುತ್ತೆ? ಒಂದು ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಚೈನ್ ಏನಾಗಿರುತ್ತೆ ಈ ರೀತಿಯಾಗಿ 5 ಪ್ರೈಮ್ ಟು 3 ಪ್ರೈಮ್ ಈ ರೀತಿಯಾಗಿ ಅರೇಂಜ್ ಆಗಿರುತ್ತೆ. ಇನ್ನೊಂದು 3 ಪ್ರೈಮ್ ಟು 5 ಪ್ರೈಮ್ ಈ ರೀತಿಯಾಗಿ ಅರೇಂಜ್ ಆಗಿರುತ್ತೆ. ಆಂಟಿ ಪ್ಯಾರಲಲ್ ಆಗಿ ಅರೇಂಜ್ ಆಗಿರುತ್ತೆ. ಓಕೆ. A purine comes opposite to the pyrimidine. Purines, adenine. So ಯಾವುದಕ್ಕೆ ಬಾಂಡ್ ಆಗಿರುತ್ತೆ? ಥೈಮಿನ್ಗೆ ಅಲ್ವಾ? That is the pyrimidine. This generates approximately uniform distance between the two strands of the helix. The two chains are coiled in right-handed fashion. So the DNA, so ಇಲ್ಲಿ ಈ ರೀತಿಯಾಗಿ ನಾನು ಹೇಳಿದ್ದೆ ಅಲ್ವಾ, ಫಾರ್ ಯುವರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್. ಸೊ ಆಕ್ಚುಲಿ DNA is coiled right handedly. ನೋಡಿ ಇಲ್ಲಿ ಏನಾಗಿದೆ ಅಂದ್ರೆ ಎರಡು ಸ್ಟ್ಯಾಂಡ್ ನೋಡಿ, ಇದು ಒಂದು ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ವಾ. ಸೊ ಈ ರೀತಿಯಾಗಿ ಕಾಯಿಲ್ ಆಗಿರುತ್ತೆ. ನೀವು ಕಾಮನ್ ಆಗಿ DNA ಡ್ರಾ ಮಾಡೋದಾದ್ರೆ ಹೇಗೆ ಡ್ರಾ ಮಾಡಬೇಕು? ಈ ರೀತಿಯಾಗಿ, ಇಲ್ಲಿ ಡೈಗ್ರಾಮ್ ಇದೆ ಅಲ್ವಾ, ಈ ರೀತಿಯಾಗಿ ಡ್ರಾ ಮಾಡಬೇಕು. ದಟ್ ಈಸ್ ದ DNA is coiled in a right-handed fashion. The pitch of the helix, so that is called as a helix anta ಅದಕ್ಕೆ ಕರೀತಾರೆ. The pitch of the helix is 3.4 nanometer or 34 angstrom. So this pitch is, the pitch of the helix is 3.4 or 34 angstrom. There are roughly 10 base pairs in turn. So ಇಲ್ಲಿ ಇನ್ ಬಿಟ್ವೀನ್ ಈಚ್ ಪಿಚ್, ಇದು ಈ ಕಾಯಿಲ್ ಇದೆ ಅಲ್ವಾ, ಇಲ್ಲಿ ಟೋಟಲ್ ಎಷ್ಟು ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ? ಬೇಸ್ ಪೇರ್ಸ್ ಮೀನ್ಸ್ ದಟ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಪೇರ್ಸ್ ಅಲ್ವಾ, ಬೇಸ್ ಪೇರ್ಸ್ 1 2 3 4 5 6 7 8 9 10. Totally 10 base pairs are present in each turn. The distance between the base pairs in a helix 0.34 nanometer, very important. So in between the base pair, ಈ base pair ಮಧ್ಯ distance ಎಷ್ಟಿರುತ್ತೆ? 0.34 nanometer or 3.4 angstrom. Okay. The plane of one base pair stalks over the other in the double helix. Hydrogen bond confers the stability of the helical structure of the DNA. So very important. Hydrogen bond that confers the stability of the structure of the DNA. Ili the central dogma of flow of genetic information. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? DNA that convert into RNA then into protein. So that is the central dogma of genetic information. So this is the very important silent features of DNA. ಇಲ್ಲಿ ಬೇಸ್ ಪೇರ್ಸ್ ಮಧ್ಯೆ ಎಷ್ಟು ಗ್ಯಾಪ್ ಇರುತ್ತೆ ನಿಮ್ಮ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ಸ್ಗೆ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಅದ್ರ ಮೇಲೆ ನಿಮ್ಗೆ DNA ಲೆಂತ್ ಅನ್ನ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಹೇಳ್ತಾರೆ. ದ ಲೆಂತ್ ಆಫ್ ದ DNA, ದ ಬೇಸ್ ಪೇರ್ಸ್ ಎಷ್ಟಿದೆ ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಅಲ್ವಾ. ಸೊ ಆ ಒಂದು ಡೇಟಾ ಮೇಲೆ ನಿಮ್ಗೆ DNA ಲೆಂತ್ ಅನ್ನ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡುವಂತ ಒಂದು ಕ್ವಶನ್ ಅನ್ನ ಕೇಳ್ತಾರೆ. ಸೊ ಈ ಒಂದು ನಂಬರ್ಸ್ ಏನಿದೆ ಅಲ್ವಾ, ಎಷ್ಟು ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ, ಅದರ ಇನ್ ಬಿಟ್ವೀನ್ ಲೆಂತ್ ಎಷ್ಟು, ಅದನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡ್ಕೊಂಡಿರಿ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಯಾವ ರೀತಿಯಾಗಿ DNA, two strands of DNA are held together. So this is the phosphate group. Here this is the pentose sugar and this is the cytosine. Okay, this is the ನೈಟ್ರೋಜನ್ ಬೇಸ್. ನೈಟ್ರೋಜನ್ ಬೇಸ್ ನೋಡಿ 3 ಬಾಂಡ್ ಇದೆ. 3 ಬಾಂಡ್ ಜೊತೆ ಇನ್ನೊಂದು ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಜೊತೆ ಅದು ಪ್ಯೂರಿನ್ಸ್ ಪಿರಿಮಿಡಿನ್ಸ್ ಜೊತೆ ಗ್ವಾನಿನ್ ಅಂಡ್ ಸೈಟೋಸಿನ್. ಇದು ಪ್ಯೂರಿನ್ಸ್, ಇದು ಪಿರಿಮಿಡಿನ್ಸ್ ಅಲ್ವಾ. ಸೊ 3 ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಜೊತೆ ಬಾಂಡ್ ಆಗಿದೆ. ದೆನ್ ಹಿಯರ್ ದಿಸ್ ಇಸ್ ದ ಶುಗರ್ ಅಂಡ್ ದಿಸ್ ಇಸ್ ದ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್. And again this phosphate group is connected to the another nucleotide ಅಲ್ವಾ, ಇದು ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಚೈನ್ ಅಲ್ವಾ. ಸೊ ಈ ರೀತಿಯಾಗಿ ಏನಾಗಿರುತ್ತೆ, ಅಡಿನೈನ್ ಮತ್ತೆ ಥೈಮಿನ್ ಇರ್ಬೋದು, ಗ್ವಾನಿನ್ ಮತ್ತೆ ಸೈಟೋಸಿನ್ ಇರ್ಬೋದು, ಕನೆಕ್ಟ್ ಆಗಿರುತ್ತೆ. ಸೊ ಇಫ್ ಯು ಅಬ್ಸರ್ವ್ ದಿಸ್ ಡಯಾಗ್ರಾಮ್ ಯು ವಿಲ್ ಗೆಟ್ ದ ಕಂಪ್ಲೀಟ್ ಪಿಕ್ಚರ್ ಆಫ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ DNA. I hope this concept is clear. Packaging of DNA helix. You know that DNA is present inside the nucleus, and how it is packaged inside the nucleus, how it is arranged inside the nucleus. Here, the distance between two conjugative base pairs is 3.4 nanometer. The length of the DNA in typical mammalian cell, that is 6.6 into 10 raised to 9 base pairs are there. 6.6 into 10 raised to 9 base pairs in diploid cell, okay, not in haploid cell. So 0.34 into 10 raised to minus 9, that is nanometer means 10 raised to minus 9 alva. So idanna multiply madre, so about 2.2 meters the length of the DNA he kandididru. Total ಎಷ್ಟು ಬೇಸ್ ಪೇರ್ಸ್ ಇದೆ, ಸೊ ಬೇಸ್ ಪೇರ್ಸ್ ಎಷ್ಟಿದೆ, 6.6 into 10 raised to 9 ಇಂಟು ಬೇಸ್ ಪೇರ್ ಮಧ್ಯೆ ಇರುವಂತ ಡಿಸ್ಟೆನ್ಸ್ ಏನು 0.34 into 10 raised to minus 9. So around 2.2 meter. How this DNA is packaged in prokaryotes. So what you mean by prokaryotes? Where the nucleus is not well defined. Nuclear region is not bounded by the nuclear membrane. So they are called as prokaryotes. They do not have definite nucleus. The DNA is scattered throughout the cell, and DNA is held together with some proteins in region called as a nucleoid. Here, in case of the in case of the prokaryotes, DNA that is packaged with the some proteins that is called as the nucleoid. The DNA in nucleoid is organized in large loops held with the proteins. That is how the packaging of DNA in prokaryotes. So this is the nucleoid region in bacteria. So very important thing is packaging in eukaryotes. In eukaryotes, the packaging is more complex. So compared to the prokaryotes, the packaging of DNA inside the nucleus. Nucleus ಅಲ್ಲಿ ಯಾವ ರೀತಿಯಾಗಿ ಪ್ಯಾಕೇಜ್ ಆಗಿದೆ DNA ಅನ್ನೋದನ್ನ ಸ್ಟಡಿ ಮಾಡಬೇಕು ಅಲ್ವಾ. There is a set of positively charged basic proteins called histones. Inside the nucleus, there are positively charged proteins. These proteins are called as histones, and this DNA is negatively charged, right? So oppositely charged molecules, they attract each other. So histones are positively charged due to the rich in basic amino acids like lysine and arginine. These proteins, they have the lysine and arginine amino acids, so they are positively charged. Histones are organized to form unit in 8 molecules called histone octamers. So histones are like this, okay, they are 8 molecules, they are held together in 8 molecules, so that is called as the histone octamer. Negatively charged DNA wrapped around the positively charged histone octamer to form the structure nucleosome. So just imagine this is the histone protein, so that is histone octamer, and negatively charged DNA that is wrapped around this protein, that is together called as the nucleosome. Enagide, this is the protein, just imagine this is the histone octamer protein. So negatively charged DNA that is wrapped around this, so idra sutta enagide suttkondige, so that structure is called as the nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. why DNA is negatively charged. Okay, so it has some negatively charged particles. Okay, so DNA ಕೆಲವೊಂದು negatively charged particles ಇದೆ. So that is the acidic ಅಲ್ವಾ. ಸೊ ಅದಕ್ಕೆ DNA negatively charge ಅನ್ನು ಹೊಂದಿದೆ. Nucleosome constitutes the repeating unit of structure in nucleus called chromatin, the thread-like stained body seen in the nucleus. So you can see, just imagine this is the nucleosome. Okay, these are the nucleosomes. Just imagine this is the DNA. So here DNA helix is formed like this, and again around this protein, again around this. So like this, the DNA wrapped around the histone proteins, and it looks, so totally it is called as a chromatin. Okay, ಈ ಒಂದು ಥ್ರೆಡ್ ಲೈಕ್ ಈ ತರ ಕಾಣಿಸುತ್ತೆ ಅಲ್ವಾ, ಅದನ್ನ chromatin ಅಂತ ಕರೀತಾರೆ together. Yes, here you can see. So this is the DNA. Okay, ಇಲ್ಲಿ ನೋಡಿ ಇದು ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್. ಈ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಸುತ್ತ ಏನಾಗಿದೆ, DNA ಸುತ್ತಕೊಂಡಿದೆ ಅಲ್ವಾ. ನೆಕ್ಸ್ಟ್ ನೋಡಿ ಇನ್ನೊಂದು ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಸುತ್ತ ಸುತ್ತಕೊಂಡಿದೆ. ಸೊ ಇನ್ನೊಂದು ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಸುತ್ತ ಸುತ್ತಕೊಂಡಿದೆ. ದಿಸ್ ಸ್ಟ್ರಕ್ಚರ್ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ chromatin network. Okay, it looks like a beads on a string. ಇದನ್ನ ನಾವು ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ನೋಡಿದಾಗ ಯಾವ ತರ ಕಾಣಿಸುತ್ತೆ ಅಂದ್ರೆ ಈ ತರ beads on the string. ಸೊ ಈ ತರ ಬೀಡ್ಸ್, ಸ್ಟ್ರಿಂಗಿಗೆ ಬೀಡ್ಸ್ ಅಟ್ಯಾಚ್ ಆಗಿದ್ರೆ ಯಾವ ತರ ಕಾಣಿಸುತ್ತೆ ಅಲ್ವಾ, ಈ ತರ ಕಾಣಿಸುತ್ತೆ. ದಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ chromatin. The nucleosome are seen as beads on string. Just ಹೇಳಿದೆ ಅಲ್ವಾ. ಸೊ ಈ ರೀತಿಯಾಗಿ this is the nucleosome around the, so like this, it is seen like this, beads on string structure when viewed under the electron microscope. The chromatin is packaged to form chromatin fibers that are further coiled and condensed in metaphase state to form the chromosome. Again this chromatin network ಏನಾಗುತ್ತೆ, ಮೆಟಾಫೇಸ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ chromatin network, it will undergo condensation and form the chromosome ಅಲ್ವಾ. ಈ chromatin network ಏನ್ ಮಾಡುತ್ತೆ, chromosome ಅನ್ನ ಫಾರ್ಮ್ ಮಾಡುತ್ತೆ ಸೆಲ್ ಡಿವಿಷನ್ ಟೈಮ್ ಅಲ್ಲಿ. Packaging at higher level required additional set of proteins called non-histone chromosomal proteins. NHC proteins are required for the additional set of higher level of arrangement. In typical nucleus, some loosely coiled regions of chromatin, so that is lightly stained, called euchromatin. Very important. So ಈ ಕ್ರೊಮ್ಯಾಟಿನ್ ಈ ರೀತಿಯಾಗಿದೆ ಅಲ್ವಾ, ಈ ರೀತಿಯ ಕ್ರೊಮ್ಯಾಟಿನ್ ಇದೆ ಅಂದ್ರೆ, ಸೊ ಲೈಟ್ ಆಗಿ ಸ್ಟೇನ್ ಆಗಿರುವಂತದ್ದು, ದೆನ್ ಲೂಸ್ಲಿ ಅರೇಂಜ್ಡ್ regions, that is called as the euchromatin. The chromatin that is more densely packed and stains dark are called heterochromatin. Euchromatin is transcriptionally active and heterochromatin is inactive. So idanna nimge keltare, write the difference between the heterochromatin and the euchromatin. What is NHC protein? Non-histone chromosomal proteins, NHC protein. Then very important, heterochromatin and the euchromatin. That is very important. That is about the packaging in eukaryotes, packaging of DNA in eukaryotes. So you can see the video.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell.
So this is how DNA is.
The process starts when DNA is wrapped around special protein molecules called
histones. The protein molecule.
The combined loop of DNA and protein is called a nucleosome.
Yes, this is the nucleosome. Okay. DNA that is
the nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here.
So again this condensed to form the chromosomes. So this chromatin network,
the end result is that the DNA is tightly packed into the familiar structures we can see through a microscope. chromosomes. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
So next topic is the search of genetic material. How they confirm that DNA is the genetic material. So many experiments were done by the scientists. So here few experiments we are studying how the DNA is considered as a genetic material. Okay. The first one is the Griffith experiment. Very important experiments for your exam. That is Griffith experiment in 1928. So his experiment based on the Streptococcus pneumoniae caused pneumonia. So there is change in the physical form of bacteria. There are two colonies of bacteria. So this Streptococcus pneumoniae, there are two colonies of bacteria. Smooth, just imagine this is the one type of bacteria, that is the smooth shiny colonies are called S strain. S strain bacteria which forms the smooth and shiny colony, and another one is the R strain bacteria that forms the rough colonies. Okay, so this is the two types of the bacteria. S strain bacteria have the mucus, that is the polysaccharide coat, and R strain does not have the mucus polysaccharide coat. That is the difference between the S strain bacteria and the R strain bacteria. S strain is virulent and caused the pneumonia in mice and died when it is infected. When this S strain bacteria, just imagine, this is the S strain bacteria. Okay, so which is injected to the mice, that mice died because of the pneumonia. Okay, so that is died. I will write like this, because of the this infection. And R strain bacteria infected to the mice, then so this is the non-virulent strain bacteria. Okay, so does not cause the pneumonia in mice when infected. It does not cause the infection to the mice, and so the mice, they are living. They do not develop any disease and they are living. They do not die. Okay. Then heat killed S strain bacteria. So the heat killed S strain bacteria is injected to the mice. Just see the experiment, very important. The heat killed S strain bacteria is injected to the mice. So when it is heat killed bacteria, it does not cause the diseases. Okay, so there is no diseases. So the mice, they are living. Then heat killed S strain bacteria S strain bacteria plus R strain bacteria, living R strain bacteria. Okay, heat killed S strain. So only heat killed S strain bacteria is injected. So there is no disease developed. But heat killed S strain plus R strain, so that is living R strain, is injected to the mice. Then mice developed the pneumonia and died because of that disease. Okay, so he recovered S strain bacteria from the dead mice. So if he observed the dead mice and recover the some S strain bacteria are developed in this. But here only heat killed bacteria are added to the mice, then they are living, so there is no disease developed in the mice. But heat killed S plus R strain is added, then they developed the disease and died. So this experiment is here. What he concluded in that experiment, he concluded that R strain bacteria had somehow been transferred, so by the heat killed bacteria, S strain bacteria. So there is something transferred from S strain to R strain. Okay, so something S strain, heat killed S strain, but it transferred something to the R strain. So the mice developed the disease, but he don't know exact what the thing is transferred to the R strain bacteria. So it is called as the transforming principle, means the heat killed S-strain bacteria transferred something. So he don't know what is that something. Okay, he transferred that to the R strain. So the R strain bacteria, they become virulent, means they develop the disease. So this, he concluded that this must be due to the transfer of the genetic material. S strain bacteria transfer some genetic material to the R strain bacteria, and R strain bacteria become virulent. Okay, and that virulence, so that when it is injected to the mice, it will become, so it developed the disease. Okay, so that is what the Griffith experiment, very important for your exam. Then, so another experiments done based on this observation, Griffith experiments observation, that is biochemical characterization of transforming principle. Here, biochemical characterization of transforming principle. Here, so this experiments that is given by Oswald Avery, Colin McLeod, and Maclean McCarthy. Prior to this work, genetic material was through to be a protein. So they thought that the genetic material was thought to be a protein. The protein is the genetic material. Okay. They worked to determine the biochemical nature of the transforming principle. They thought that protein is the genetic material, but they performed the experiment. What was that experiment? They purified some biochemicals, that is proteins, DNA, RNA from the heat killed S cells to see which one could transfer live R cells to S cells. Okay. What they did? So they added S strain, that is the virulent. Okay. Plus protease enzyme. Okay, they purified the biochemicals. So just imagine here, they added the biochemicals. Okay, protein, DNA and RNA. So inside this, so just take this. So here protein plus DNA and RNA is present. Okay, so these three constituents are present. Here, same thing they have added heat killed S strain plus living R strain. So same as the Griffith, last step of the Griffith experiment, and the protease enzyme. Protease enzyme is added. Okay, so protease enzyme that digests the protein. So protein anna digest ಮಾಡಿಬಿಡುತ್ತೆ, only DNA ಮತ್ತೆ RNA ಉಳ್ಕೊಳ್ಳುತ್ತೆ. Okay, then here heat killed S strain and the living R strain. So here when it is injected to the mice is died. Okay, so means something is transferred. That means something is transferred. So again here, these three biochemicals, proteins, DNA and RNA. RNA's enzyme is added. RNA's enzyme means which digests the RNA, which digests the RNA. Okay, so here RNA is digested, only DNA and protein is transferred to the R cell. So there is transformation, means which there is a development of disease in mice. Okay, so again in the last step they added DNAs, means DNA digesting enzyme, and heat killed S strain and the living R strain. So there is no transformation. The mice they developed, there is no transformation of the genetic material, means the mice they do not develop any disease. Means they concluded that when DNA is digested, protein is present. Protein ittu, RNA ittu alva. ಸೊ ಪ್ರೋಟೀನ್ ಇತ್ತು, ಆರ್ ಎನ್ ಎ ಇತ್ತು, ಅದೇನಾಗ್ಲಿಲ್ಲ, ಟ್ರಾನ್ಸ್ಫರ್ ಆಗ್ಲಿಲ್ಲ ಅಲ್ವಾ. ಸೊ how they concluded this experiments? Proteins of heat killed S strain is not a genetic material. RNA of the heat killed S strain is the not a genetic material. DNA of the heat killed S strain is the genetic material, because DNA digested with DNAs mixed with the R strain, unable to transform the R strain to the S strain bacteria. R strain ಇಂದ S strain bacteria ಆಗಿ ಅದು ಕನ್ವರ್ಟ್ ಆಗಲಿಲ್ಲ. ಸೊ ದಿಸ್ ಇಸ್ ದ conclusion of this experiment. Very, very important. I hope this experiment is clear. So these all are the experiments that was done by the scientists to detect the DNA is the genetic material, to confirm that DNA is the genetic material. And here one more experiment that is called as Hershey-Chase experiment. So one of the very important experiment, Hershey-Chase experiment. They grew some viruses on the medium that is having the radioactive phosphorus and some on the medium having the radioactive sulfur. Okay, they grown in this experiment, Hershey-Chase grown. So this is the bacteriophage virus. So the bacteriophage virus that is grown in the radioactive just a minute, they grown in the yes, one virus that is grown in the radioactive phosphorus have the radioactive DNA but not radioactive protein, because phosphorus is present in the DNA, not in the protein. ಇಲ್ಲಿ ಏನ್ ಮಾಡಿದ್ರು ಅಂದ್ರೆ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ, so that is the bacteriophage virus, sorry not bacteria, virus. So ಅದನ್ನ ಏನ್ ಮಾಡಿದ್ರು, ಫಾಸ್ಫರಸ್, ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಫಾಸ್ಫರಸ್ ಅಲ್ಲಿ ಗ್ರೋ ಮಾಡಿದ್ರು. ಓಕೆ, ಸೊ ಒಂದು ಮೀಡಿಯಂ ಇತ್ತು, ಆ ಮೀಡಿಯಂ ಅಲ್ಲಿ ಅದು ಗ್ರೋ ಆಯ್ತು, ದಟ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ ದ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಫಾಸ್ಫರಸ್. ಸೊ ಇದ್ರ ಒಂದು ಡಿ ಎನ್ ಎಲ್ ಏನಿತ್ತು, ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿ ಎನ್ ಎ ಇತ್ತು. ವೈ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿ ಎನ್ ಎ ಇಸ್ ಪ್ರೆಸೆಂಟ್? ಸೊ ಡಿ ಎನ್ ಎ ಅಲ್ಲಿ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಫಾಸ್ಫರಸ್ ಇದೆ ಅಲ್ವಾ, ಡಿ ಎನ್ ಎ ಅಲ್ಲಿ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ಇದೆ ಅಲ್ವಾ. ಸೊ ದಟ್ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ದಟ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ ದ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಕಂಟೆಂಟ್. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ಇಲ್ಲಿ ಡಿ ಎನ್ ಎ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಆಗಿತ್ತು, ಬಿಕಾಸ್ ಇಟ್ ಕಂಟೈನ್ಸ್ ಇಟ್ ಡೆವಲಪ್ಡ್ ಇನ್ ದ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಫಾಸ್ಫೇಟ್ ಅಲ್ಲಿ ಗ್ರೋ ಆಗಿತ್ತು. ಅದಕ್ಕೆ ಇದರ ಡಿ ಎನ್ ಎಲ್ ಏನಿತ್ತು, ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಫಾಸ್ಫೇಟ್ ಇತ್ತು. ಸೊ ದ ಡಿ ಎನ್ ಎ ಇಸ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿ ಎನ್ ಎ. ಅನದರ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ದಟ್ ಈಸ್ ಗ್ರೋನ್ ಇನ್ ದ ಅನದರ್ ಮೀಡಿಯಂ. ಓಕೆ, ಸೊ ಇನ್ನೊಂದು ಮೀಡಿಯಂ ಅಲ್ಲಿ ಗ್ರೋ ಆಗಿತ್ತು, ದಟ್ ಈಸ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಸಲ್ಫರ್ ಮೀಡಿಯಂ ಅಲ್ಲಿ ಗ್ರೋ ಆಗಿತ್ತು. ಸೊ ದಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ದಟ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ ದ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ದಿಸ್ ಪ್ರೋಟೀನ್ ಕ್ಯಾಪ್ಸೂಲ್, ಈ ಕ್ಯಾಪ್ಸೂಲ್ ಏನಿದೆ ಅಲ್ವಾ, ಈ ಕ್ಯಾಪ್ಸೂಲ್ ಮಾತ್ರ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಆಗಿತ್ತು, ಈ DNA ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಆಗಿರ್ಲಿಲ್ಲ. ಸೊ ಯಾಕೆ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಆಗಿರ್ಲಿಲ್ಲ ಅಂದ್ರೆ, ದ ಸಲ್ಫರ್ ದಟ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ಪ್ರೋಟೀನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಬಟ್ ಇಟ್ ಈಸ್ ನಾಟ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ DNA. ಅದರಿಂದ ಈ ಪ್ರೋಟೀನ್ ಕೋಟ್ ಮಾತ್ರ ಏನಾಗಿತ್ತು, ದಟ್ ಈಸ್ ಬಿಕಮ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್. ಓಕೆ, so when these viruses are infected to the bacteria, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಗೆ ಇನ್ಫೆಕ್ಟ್ ಮಾಡಿತ್ತು ನೋಡಿ. ಸೊ when they are infected to the bacteria, they injected their genetic material. Okay, genetic material ಅನ್ನ ಇಂಜೆಕ್ಟ್ ಮಾಡ್ತು ಅಲ್ವಾ. ಸೊ this bacteria also injected the genetic material. So this genetic material that is blending with the genetic material present already in the ಬ್ಯಾಕ್ಟೀರಿಯಾ ಜೊತೆ ಇರುವಂತಹ ಒಂದು ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಜೊತೆ ಬ್ಲೆಂಡ್ ಆಯ್ತು ಅಲ್ವಾ. ದೆನ್ ನೆಕ್ಸ್ಟ್ ಸ್ಟೆಪ್ ಅಲ್ಲಿ ನೋಡಿ, centrifugation. The virus particles were separated from the bacteria by spinning them in a centrifuge machine. Okay, so ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅನ್ನ ಏನ್ ಮಾಡಿದ್ರು, ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡಿದ್ರು. ದಟ್ ಈಸ್ ದ ಮೆಥಡ್, ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮೆಥಡ್. ಏನಾಗತ್ತೆ, ಹೈ ವೇಟ್ ಇರೋದು ಬೇಸ್ ಅಲ್ಲಿ ಇರುತ್ತೆ ಅಂಡ್ ಇನ್ ಸ್ವಲ್ಪ ಏನಾಗಿರುತ್ತೆ, ಸೊ ದಟ್ ಈಸ್ ಅಟ್ ದ ಟಾಪ್. ಓಕೆ, ಸೊ ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡಿದಾಗ ಏನಾಯ್ತು ಹಿಯರ್, ಸೊ ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡಿದ್ರು ಇಲ್ಲಿ. ಸೊ ಇನ್ ದಿಸ್ ನೋ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೆಲ್ಸ್. ಈ ಸೆಲ್ಸ್ ಒಳಗಡೆ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಸೆಲ್ಸ್ ಇರ್ಲಿಲ್ಲ, ಬಿಕಾಸ್ ಸೊ ಇಟ್ ಇಂಜೆಕ್ಟೆಡ್ ಇಟ್ಸ್ ಓನ್ಲಿ ಏನಾಗಿತ್ತು, ಈ ಇದನ್ನ ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡಿದಾಗ ಏನಾಯ್ತು, ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ನಲ್ಲಿ ನೋ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೆಲ್ಸ್ ಒಳಗಡೆ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಇರಲಿಲ್ಲ, ಬಟ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೂಪರ್ನಾಟೆಂಟ್ ಅಂದ್ರೆ ಮೇಲ್ಗಡೆ ಇರುವಂತಹ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಅಲ್ಲಿ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇತ್ತು, ಸೆಲ್ಸ್ ಒಳಗಡೆ ಇರಲಿಲ್ಲ. ಬಟ್ ಹಿಯರ್ ಇನ್ ಕೇಸ್ ಆಫ್ ದಿಸ್ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೆಲ್ಸ್ ಒಳಗಡೆ ಏನಿತ್ತು, ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇತ್ತು, ಬಟ್ ನೋ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೂಪರ್ನಾಟೆಂಟ್ ಒಳಗಡೆ. ಇಲ್ಲಿ ಮೇಲ್ಗಡೆ ಏನಿತ್ತು ಅಲ್ವಾ, ಇಲ್ಲಿ ಮೇಲ್ಗಡೆ ಏನಿದೆ, ಪ್ರೋಟೀನ್ ಕೋಟ್ ಇತ್ತು. ಪ್ರೋಟೀನ್ ಕೋಟ್ ಅಲ್ಲಿ ಏನಿದೆ, ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಇದೆ ಅಲ್ವಾ. ಸೊ ಇನ್ ದ ಸೂಪರ್ನಾಟೆಂಟ್ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇಸ್ ಡಿಟೆಕ್ಟೆಡ್, ಬಟ್ ಹಿಯರ್ ಇನ್ ಕೇಸ್ ಆಫ್ ದಿಸ್ ದ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಡಿಟೆಕ್ಟೆಡ್ ಇನ್ ದ ಸೆಲ್ಸ್ ಒಳಗಡೆನೆ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇತ್ತು. ಸೊ ದಟ್ ಈಸ್ ದ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಡನ್ ಬೈ ದ ಹರ್ಷೆ ಅಂಡ್ ಚೇಸ್. ಹಿಯರ್ ವಾಟ್ ದೇ ಅಬ್ಸರ್ವ್. So in this experiment, what they observation, so that is bacteria infected with viruses that had to radioactive DNA were radioactive and no radioactivity present in the supernatant. So observation is very important. Illi nodi, virus that had radioactive DNA ididenaagittu, so radioactivity present in the cells, not in the supernatant. Bacteria infected with viruses that had radioactive protein were not radioactive, but radioactively found in the supernatant. Okay, so what is the conclusion of this experiment? DNA is infecting agent that made the bacteria radioactive. Hence DNA is the genetic material, not the protein genetic material alla anta. So this experiment is proved. Okay, so that is the Hershey-Chase experiment. Very important, Hershey-Chase experiment. Then, so if you compare the DNA and RNA, DNA versus RNA. So what are the characteristics of RNA and DNA? Why DNA is prepared as a genetic material, so not RNA. Okay, here RNA is unstable, so it is not stable. Why RNA is not stable? Because at the 2' end OH group is present at every nucleotide in the RNA is reactive group and makes RNA liable and easily degradable. And RNA is also not known as a is also known as the catalyst, hence it is reactive. Then RNA is unstable and mutates faster. Consequently, the viruses having RNA genome and having shorter lifespan mutate and evolve faster. So that is having the mutation capacity. Then a biomolecule must show the some why genetic RNA and DNA are the genetic materials. Here, so the genetic materials that must show some specific characteristics to become a genetic material. Both the nucleic acids can direct their replication unlike the biomolecules in a cell. RNA is not a very suitable genetic material. What is the reason behind this? Here, the ribose sugar of every nucleotide in RNA consists of reactive 2 prime. So at the second carbon atom, hydroxyl group is present. Alva. And RNA also functions as enzymes in ribosomes, thus making unstable and reactive. And uracil is present in RNA is less stable than the thymine of the DNA. So uracil enilla, so that is less stable compared to the DNA. Okay. And RNA single stranded. The nitrogen bases in RNA can easily bind with the other molecule by forming the hydrogen bond. So RNA ಏನಾಗ್ಬಿಡುತ್ತೆ ಇನ್ನೊಂದು ಮಾಲಿಕ್ಯೂಲ್ ಜೊತೆ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್ ಮಾಡ್ಕೊಂಡು ಏನಾಗ್ಬಿಡುತ್ತೆ ಬೈಂಡ್ ಆಗ್ಬಿಡುತ್ತೆ. ಸೊ ಅನ್ಸ್ಟೇಬಲ್ ಅಂಡ್ ರಿಯಾಕ್ಟಿವ್ ಇನ್ ನೇಚರ್. ಸೊ RNA ಅನ್ಸ್ಟೇಬಲ್ ಇದೆ ಯಾಕೆ and it is reactive. Being unstable, RNA mutates at faster rate. RNA mutation, so it will undergo mutation, change ಆಗತ್ತೆ. Such fast changes are harmful in higher life forms. So it is harmful for the higher life forms. And why DNA is genetic material in most of the organisms? So DNA yake genetic material in case of higher organisms, that we learn in the next class. Okay, I hope this class is very useful for you people. I hope you enjoyed the class. Just I will I'm going to stop the class here. In the next class, I will continue. Okay. So if you have any doubts, please write down in the comment box. Thank you.